pepsin, and pancreatic carboxypeptidase. All right, so these first few enzymes are involved in protein digestion. We started the protein digestion in the stomach using pepsin. Now we're going to complete enzymatic hydrolysis of these polypeptide chunks using more proteases. These are collectively protease enzymes because they cleave peptide bonds within polypeptides. Okay? Now, we also have another group of enzymes here called pancreatic nucleases. And collectively as a group, the pancreatic nucleases are involved in hydrolyzing nucleic acid down into the individual nucleotide monomers. Okay? So that should get us to the nitrogenous bases, plus the phosphates as a separate piece. And then there's also some pancreatic lipase. These enzymes facilitate the separation of the glycerol from the fatty acid chains, because those were put together through dehydration synthesis. So these lipases can do that hydrolysis of triacylglycerides. Okay? And then the last group here, the pancreatic amylases. There was a salivary amylase that began carbohydrate digestion in the mouth, in the oral cavity. This one, the pancreatic amylase, is going to complete digestion of the carbohydrates. It's maltases, right? We're cleaving apart the alpha-1,4 linkages between the two alpha glucoses that make up maltose. Okay? All right. So the pancreas is producing a lot of these enzymes that are necessary for hydrolysis within the small intestine. And the idea is if we do a good job of this, what should be produced in the small intestine is a bunch of monomers. Right? We began carbohydrate digestion in the mouth, we completed it in the small intestine. We began protein digestion in the stomach, we complete it in the small intestine. We begin digestion of lipids, specifically triacylglycerides, and nucleic acids within the small intestine. Okay? All right, the pancreas also produces a very, very important alkaline solution. All right, this is a really, really important one. Lots and lots of bicarbonate are dumped into the small intestine. The idea is that acid chyme still has a very low pH. So as it hits the small intestine, if we don't raise the pH, the acid chyme will begin to digest the material that makes up the lining of the small intestine. And we don't want that. We're going to end up with an ulcer in the small intestine. So this bicarbonate, what it does is it neutralizes the acid chyme. It brings the pH back to a reasonable value, and it's that value that all these digestive enzymes work properly at. Okay? All right. Let's go back up here for a second. The next one of these, the liver. The liver's function here is to produce a substance called bile. Now, bile is not an enzyme. So I'll put that, not an enzyme. Okay? Bile is a material. It works a lot like a detergent. It's an amphipathic molecule, specifically an emulsifier. And what emulsifiers do and detergents is take nonpolar molecules and polar molecules and evenly distribute them throughout a liquid. So for example, if you were to take milk and put it in a container fresh out of the cow, the milk would begin to separate. There would be a bunch of fatty stuff at the top, the hydrophobic molecules, and a bunch of this liquid stuff here at the bottom, which is the hydrophilic molecules. So when we take milk out of the cow, we add an emulsifier to it. And what that emulsifier does is evenly distribute everything together so that you don't get this separation of fat and water. We refer to that as homogenation, right? Homogenized milk, but it's basically this emulsifier that's added that does the homogenizing. So bile is an emulsifier. That means when it hits the small intestine, it's going to take all of the lipids that you've taken into your diet and evenly distribute them with everything else so that we don't end up with these big globs of lipid moving through the digestive system. It also allows enzymes to get at them easily. If enzymes, the lipases, have hydrophobic regions on them, it would be difficult for them to interact with fats if we didn't emulsify everything, right? If we didn't bring everything together, because they'd be hanging out in two different parts of solution. Okay? Now, the liver, when it produces the bile, some can move directly to the small intestine, but a lot of it gets moved to the gallbladder that does storage of bile. The gallbladder doesn't produce anything. All it does is store this bile. Okay? You doing okay so far? So the duodenum, this first part, this is where a lot of this stuff is happening. We're doing a lot of digestion here in the lumen of the small intestine, breaking things down into smaller and smaller pieces. Okay? Once we've broken things down, we've done the digestion, the next step would be absorption. Okay? All right. So in order to do absorption properly, what we need to do is move materials from the lumen of the intestine into the cells that line the small intestine. Because after all, the lumen of the small intestine, as I've mentioned before, is technically outside of you, right? All this digestion is extracellular. We need to bring that material through absorption into cells. So if we look at the lining of the small intestine here, we would see that there are lots and lots of folds here. That would be the lining of the small intestine. That's the lumen of the small intestine here. And then if we zoomed in on this, we would see this kind of an effect. So really, if I was going to draw this the way it really looks microscopically, it would look like this. That makes sense? So in our structure here, this kind of stuff is called mucosa. That's the lining of the small intestine. But if we zoomed in on this stuff and we're looking at these tiny little projections off of the mucosa, this projection is called a villus. So that means in our figure what we've done is taken a larger structure, look at it under a higher power lens on the microscope, and then under a still higher power. So here we're looking at a little tiny portion showing that there's tremendous amounts of surface area. The more surface area, the more surface for absorption. So there's a certain efficiency that you gain by having additional surface area to allow for this absorption. Okay? All right. Now, the villi, if I looked at a villus, it also has projections off of it as well, and I can't really draw that easily, but the idea is lots and lots and lots of surface area. Tremendous amounts of surface area for absorption. Okay? All right. Let's look at an individual villus. So here, that's an individual villus. Okay? That gets us to this individual villus. Okay? Now, if we look at how this is arranged, there should be a layer of epithelium here. Just check the focus a little bit. All right, so this is an epithelium here. And then within that surface area exposed to the lumen, there's capillary. So one side of the capillary would be the venous system coming in, and the other side of the capillary is the arterial system coming out. So my expectation is there would be lots of locations along here where the capillary is spread out. Okay? Then the idea is the microvilli here, the surface epithelium of that microvillus pulls material in, and then ultimately we're going to try to move stuff into the capillaries so that the material that's been absorbed from the small intestine ends up in the circulatory system. Okay? Now, we're going to be able to do that with everything that's water soluble, because those materials can enter into the fenestrated capillaries, and they will be carried in solution through the circulatory system. So that should cover 3 of those biological molecules: amino acids, nucleotides, and sugars, right, the monomer sugars. Those can be moved through the circulatory system. And the idea is these different capillary networks associated with each of the microvilli, they begin to join together, and you would get larger and larger and larger and larger systems coming together. I think I mentioned this backwards. I was trying to look at this and talk at the same time. So here is an artery, right? This should be then the vein side of things. I've got the blood moving in the wrong direction based on the figure, right? Blood would come in this way, right across through here too, and would exit on the venous side. Sorry about that. Then we would collect these individual venules, right, together and together and together until all of these venules coming from all of the capillary beds of all of the microvilli come together in one large vein. This vein is carrying all of the nutrients that were picked up from the small intestine as the blood flowed through these capillaries. Okay? This one vein is called the hepatic portal vein. The hepatic portal vein. This vein, hepatic, think liver, this one vein is headed to the liver. Okay? What the liver will do is all of this loads and loads of monomer nutrients that have come from the circulatory system through the hepatic portal vein, the liver is going to make a lot of adjustments to the levels of these nutrients. Take a bunch of amino acids out of solution, build albumin, put it back in. Take a bunch of sugar out, build glycogen, and only release it with the appropriate signal from the pancreas. take a lot of those nucleotides out, dole them out separately. So the liver in this case is functioning as kind of a nutrient filter to filter out as many nutrients as we can so that we keep homeostasis with respect to the levels of these nutrients in the circulatory system. Okay? So the liver is other function here that's kind of non-digestive, is regulating the levels of all these nutrients after they come out of the small intestine. Okay? All right, now, in terms of moving things into these capillaries, some of this movement is diffusion related because I have a higher concentration, sorry about that, because I have a higher concentration of nutrients here than in the arterioles coming into the capillaries, in which case I'm moving down a diffusion gradient. That's things like fructose. Fructose moves down a diffusion gradient through that fenestration and moves in. Others, like amino acids, have to be actively transported into the capillaries. Okay? Probably because they're much larger molecules. Okay? All right, now, that covers carbohydrate, protein, nucleic acid, right? We still have one group left, and that's the lipids. Well, the lipids don't want to be solubilized in the blood serum. It's just not going to happen. They don't want to do that. And it would be kind of risky to dump a bunch of this lipid directly into these and then collect all of that lipid and then move all of that lipid to the hepatic portal vein. The problem is all this lipid would be coming all at once, and we get these big lipid blobs, and that's just not going to work very well. So what we're going to do is have another network of tubes. We'll run one of these tubes up through here, right here, and this tube is called a lacteal. Right? In the diagram above, it's in kind of that cyan color, kind of a bluish green. That's a lacteal. The lacteal is connected to a network of tubes that together constitutes the lymphatic system. We'll talk lymphatic system when we get to the immune system, but for right now, it's a separate